this is Dr. Mineza, and you are listening to the second lecture of my course Research Methodology. And in this lecture, we're going to cover some basic stages of a research project life cycle. And it is designed in such a way that will help you identify the loopholes in your study, which is currently underway or you are planning to publish. Recalling you the significance of having good observation in the field of research, Newton's law of motion, and if I recall you, it all began with a simple observation of apples falling from the tree, and Newton looked at it from the eyes of a physicist. So I've included the quote over here of Robert Greene. He says, "Reason, observation, and experience, the holy trinity of science." And be it a computational chemist or any field of engineering, there are many important physical relations that you derive from basic laws of motion that were proposed by Newton. So being a keen observer really helps if you are into the field of research. Throw back to the first lecture, and I told you guys about the future of those who want to be in the field of research. So this is from the first lecture, and I told you that research motivation is actually a function of your aptitude and willingness. So those of you who can see a problem and think they can find a solution, and those of you who see a problem as an opportunity, the smart guys who drive the knowledge economy, and those of you who see a problem but they don't have any idea of finding solution, this course is for them. And they all have one common characteristics: their ability to identify a problem. That is their keen observer. And it's mainly because you have curiosity to know things, and you look at things from a different perspective, which normally people might not notice in routine life. Like falling of apple from trees, it's a routine thing which Newton observed differently. So if you're a novice and you're on your way to embark a holy journey of research study, then this course is specially designed for you. It is designed to equip you with right techniques and tools that will help you complete your research study successfully and in a very meaningful way, so that your research outcomes are beneficial for a wider community of scientists. So let's begin. These are the basic stages of any research study, be it from medical science or engineering background. And first you identify a problem, and then you perform a thorough literature search, and then you formulate your research hypothesis. You plan your study, what tools you will be using, what timeline you have to perform the study, and you collect your data, and using the tools within the time frame that you have planned. This is really important. And you collect your data, and then you perform analysis using different data analytical techniques. And once you are done interpreting your data, you report it in the form of scientific article in any international journal, or you publish it as a dissertation with your affiliations. In today's lecture, we will be focusing on problem identification, formulating concise research statement, setting up practical research goals, deciding which methodology you should be working with, and what experimental design you're gonna use for your study, considering your research goal. Starting with problem identification. The moment you see there exists a problem, your research begins. And remaining stages are all about magnifying the problem and finding solution. So during the course, during the length of this course, I'll present you two research case study so that you will grasp the basic concepts of research methodology. One is from medical science background, while the other is more of an engineering problem. It will help you go through basic steps involved in any research study and give you a different perspective. So starting with medical research problem. Those of you who are medical professionals routinely observe patients and prescribe drugs and perform follow-ups. Agree? So this implies that you got plenty of data to perform any clinical study. You will agree with me. So here I have presented a common and very well-studied medical research problem: diabetes associated obesity. And diabetes type 2 it's very common nowadays, and majority of diabetic patients, they fail to maintain BMI less than 30, and they fall in the category of obese individuals. It renders them susceptible to cardiovascular disorders. So consider here a thoughtful and concerned physician, and half of whose patient who she examined monthly are diagnosed with type 2 diabetes, and half of those who are diabetic, uh, the physician noticed that they are obese, and they develop cardiovascular disorder at the later stages of their life. So let's say a fraction of it dies cause of myocardial infarction, heart attack. So this actually or this observation uh, made a physician think, why am I obese? Diabetic patients are more prone to myocardial infarction. It means the clinician observed that there exists a problem with diabetic patients. Those who tend to be obese are more susceptible to cardiovascular disorders. So this problem can be investigated if the physician got the willingness or aptitude to conduct a whole research study on it. So this is a thoughtful clinician. We jump on to another research problem, which is more of engineering nature, particularly a biomedical engineering problem. Cardiac stenting is performed in individuals who suffer from stenosis in their coronary arteries, which are basically the blood vessels of heart. And what happens, you insert a balloon expandable metallic tube with meshwork made of either 316 stainless steel, cobalt, chromium, or nitinol, and the meshwork has got specific strut geometries and dimensions depending on the brand you are using. So a common of not so uncommon experience cardiologists have associated with cardiac stenting is deployment failure. So we consider here a case in a hospital. There are 4 cardiologists, let's say, and they perform coronary stenting using 2 different stents, let's say stunt A and B, and they both have different geometries, as you can see over here. So one of them is a slotted tube, while the other one is wire coil. So on-site fracture or stent dislodgement were observed in 50% of the performed cases. This is just a case we have made up ourselves. Being a biomedical engineer, it's your job to find out whether it's an interventional issue, manufacturing fault in the product delivered to the hospital by XYZ supplier, or is it some design related issue. Once you are done identifying the problem, a clear, concise research statement needs to be formulated. And what we saw earlier, the examples we discussed, there were 2 generic, and now you have to go through a series of steps to formulate a proper research statement so that your reader will understand the direction of your research. So these are the few general steps you're going to go through to formulate a proper research statement. After making a generic problem statement, you need to understand the nature of research you're going to do. Whether it's going to be exploratory, descriptive, empirical, or observational. So once you are done deciding the nature of your study, the next step is to see what others have done in that domain. This is really important with regard to novelty of your work. The more detailed literature searches, the more novel research design you're gonna make. So it's the rule of thumb. And seeing what others have already done, you move on to the next step. After making contextual correlation, you try to figure out something which is lacking and still needs to be answered to address the problem you just identified. So those are the loopholes that you're going to work on. And after you're done doing your extensive research and finding the loopholes, now you have a clear picture of what you want to do and in which direction you want to work on. So now it's time to rephrase your research statement. So back to the medical problem, your genetic observation was heart attacks in diabetic patient, right? So this time you want to make it more specific after digging massive literature, because you see that others have done similar kind of research studies. You rephrase your research statement, and you gonna make it like diabetes mellitus type 2 associated obesity and increased susceptibility to myocardial infarction. Now moving on to the engineering problem. Likewise, you did detailed research on cardiac stents, and you studied that strut geometry, it varies in different brains, and there are dislodgement issues associated with slotted tube stents. So your common observation before literature search was stent deployment failure, right? And you figured out that two different brands of stents, they differ in the meshwork geometry, and then after understanding the geometrical parameters that affect the performance of stunt in vivo, you rephrased your research statement and made it more refined, like strut geometry and rate of dislodgement of coronary slotted tube stents. So now it's more specific. Moving on from formulating a statement to setting up research goal. Usually after you are done formulating your research statement, your research objectives automatically become very clear. You know what you want to do. So back to medical problem, diabetes mellitus type 2 associated obesity and increased susceptibility to myocardial infarction. So we have defined some goals for it. The first one, to investigate the mortality rate of diabetic patient due to MI, shorthand of myocardial infarction. It's doable and understandable. The second one is to do the clinical profiling of the diabetic patient who are obese and susceptible to cardiovascular disorders. This is again understandable and doable. The third one is to probe the role of antidiabetics in obesity. So in the defined scenario, the first three objectives are completely doable. The last one I have highlighted is to unravel the underlying molecular mechanism that leads to heart failure in obese diabetic patients. This requires a whole another study, a different research study of different nature, different experimental design. So it's not realistic to have this research goal in the context of the medical research problem we discussed earlier. The engineering problem we talked about is strut geometry and rate of dislodgement of coronary slot tube stents. So the first three goals again are doable and understandable. I read it for you. Computational study of slotted tube geometry stent and its impact on hemodynamics. You can do it through computational fluid dynamics techniques. The second one is investigation of crack fatigue and overload fracture mechanism, slotted tube geometry. Again, you can do it. And the third one is to find the correlation between strut thickness and rate of dislodgement. You can also do it in the given scenario. But the last one, to fix the geometrical flaws in stent and clinically evaluate its performance. It seems like you're not familiar with the IRB approval process, and when you are proposing, something new or something, some modification in the current design. It's a whole lot of process. It's a different sort of research study. So you need an entirely different research design to conduct this last to achieve this last objective. So it's very unrealistic to have it in the given scenario. So from your literature search, you found that there are multiple geometrical factors which define in vivo performance of a stunt. So let me tell you something. Stunts with sharp protruding geometry after expansion, they can promote in stent restenosis. So you decided before blaming surgeons, going back to the scenario that I explained in the start, why not dusting off your computational skills and study the hemodynamics of the stunt geometry that your lab is using. There are a couple of different techniques you can use to study the fracture mechanism in different types of stunt design. And your job as a biomedical engineer ends once you find out what causes stunt fracture, and be it an interventional issue, manufacturing fault, or design fault. And guys, please, you gotta be good at human anatomy and physiology when you want to understand how these devices will perform in vivo. And also to be able to understand if your surgeon is using the device that you manufacture correctly or not. You gotta understand his job very well, right? So you can't just simply say understanding the medical part is not an engineer's problem. This is not appropriate. Remember, you are a biomedical engineer and it's an interdisciplinary field, so no excuses, please. Set objectives that will help you determine the characteristics of your data, and it will also help you understand the mode of data collection and the time frame you should pick up to gather your data, in case if it's a retrospective or prospective study. So you will pick up your time frame very carefully, and how you will be collecting data. Your research or design should be selected as per your objectives and goals, and your mode and procedure for data collection should also safeguard against bias and reliability. You gotta be careful about it. Many of us, we don't consider this factor. And use the data analytical technique that suits most to your data set. This is also important. You just can't simply use any data analytical technique to analyze the collected data and to interpret it. No, you can't simply do it. You need to understand the characteristics of the data that you have collected, and then you're going to decide which statistical method you should use to make a conclusion or inferences. Now, let's talk about methodology. Where to row your boat, right? And this is the most important part of today's lecture. So, before you start doing your research, I have repeatedly mentioned that you need to know the nature of your research. Is it going to be exploratory? Is it going to be descriptive? Is it going to be quantitative, qualitative, or fundamental? And if it's applied, I need to warn you about something. My doctoral study, it was quite interdisciplinary, and because I had a very diverse academic background, I was able to conduct it. I did my bachelor's in biotechnology, and then my master's in biomedical, and then I did my doctorate in nanoengineering. So the nature of the research I did really helped me. The academic background that I had, it really helped me in conducting the research study. So if you are from a background which is different from the type of the research that you're going to do, make sure that you acquire the skills necessary to conduct the study you are interested in. And the second part is, while deciding methodology, what relationship are you trying to establish? This is really important. You need to know your variables. And this is important because sometimes you decide to work with something which is not practical in your scenario with the provided resources, and you end up messing up all your research project. So it's always advisable to start to decide in the start what variables you want to work with and how it's going to contribute towards achieving your XYZ research goals. So you need to be crystal clear how measuring XYZ variable will help you achieve your um, XYZ goal. So the last one is how it contributes to the goals or objectives of your research study. To make it more clear, we go back to the medical research problem that I have been quoting again and again, and I will be. So you want to know the parameters that will help you determine how diabetic a patient is, how prone he or she is to obesity or cardiovascular disorders. So the variables like HbA1c levels, blood pressure, cardiogram, and lipid profiles, these are the variables which are useful in the medical research problem that I have been quoting since start. And you can make decisions using these variables if you are collecting these variables or measuring these variable during the course of your study. So it can help you decide susceptibility of diabetic patient to cardiovascular diseases, and in case if you perform drug profiling like the anti-diabetic that a clinician normally prescribe to his or her patients. So if you do profiling of the drug of diabetic patient that you want to study along with their diet routine, it can help you find the possible role of anti-diabetic in obesity. So this is about selecting your variables which can be measurable and you have the skills to measure it. This is also really important. And if you're working with applied research and you're working with variables that you might need a whole different set of skill to measure or collect data, so you need to make sure how you're going to manage it. Now I'll be talking about the importance of data analytical techniques, and I've noticed quite often researchers don't carefully select a correct statistical method to make inference on their findings. So I'll be discussing some common mistakes, and the first one I've listed over here is a subtle mismatch between the variables you're working with and selected statistical method. For example, the number and type of extraneous variables are blatantly ignored in experimental design. And instead of applying Kruskal-Wallis analysis of variance, you apply one-way ANOVA. And the second one is ignoring extraneous variables and confounded relationships that significantly affect the research outcomes of your proposed study. So it's related to the first one, and it's also about not considering external factors that affect your variable, experimental variable. So I'll give you an example over here. You design a drug, let's say drug T to treat a disease X, and drug T works by blocking a factor A in some biochemical pathway. And you ignore the fact that there exists disease Y which shows comorbidity with disease X, and its treatment is dependent on the expression level of factor A. So the applicability of the drug that you have designed is now limited because you haven't considered the extraneous factor. Disease Y is dependent, shows comorbidity with disease X, and it shows dependence on the expression levels of the factor that you were suppressing through the drug T that you just designed. So you need to consider extraneous factor. And the last one is you forgot to apply statistical data analytical technique. How come? You don't want to do it. Few other common mistakes researchers make. They apply a parametric test on non-Gaussian data. They do not run a normality test, and they, without knowing the distribution of their data, they apply a parametric test on on it. The second one is treating non-normal data as normal due to lack of biostatistics knowledge. You don't know how to run a normality test, and you just randomly select any technique to reach a conclusion. The third one is not knowing exactly the number of the control and experimental variables, and poor selection of experimental design that leads to erroneous analysis of data. So, an example that I want to give you over here is you are developing a vaccine uh, for uh, for a population not limited to some region. And while designing vaccine, you ignore factors like region-specific strains, genetic susceptibility of a population, and patients at broader ends of a clinical spectrum. You ignore them. So when you're gonna get the data of the efficacy of your vaccine, it won't show you a normal distribution because you didn't take into account the extraneous factors. Now coming to the importance of sampling, and this is another common mistakes that I normally find in some research studies. Sampling affects decision making on your research outcome. And let me tell you guys, research with n is equals to 1, that is the number of samples is equals to 1, and excellent results are simply research junk. It's meaningless. And clinical trials that report efficacy of some proposed treatment with 1 to 5% of the representative population, and ignoring the patient at a different clinical spectrum. I tell you what, the research outcomes of such clinical trials are insignificant, and it can be used in making informed decision. And this is applicable to the vaccine example that I've given you earlier. You really need to have a good sample. In hypothesis testing studies, sampling size affects confidence interval of your conclusion. And a large sample, if you select from a bigger population, it will give you statistically significant result. So you want to make sure that you get a good sample size to make an inference of your research hypothesis.